Hello everyone. In this video I'm going to be talking about stem cells. We'll talk about what they are, where they're found in the different types, but also what they can be used for. So some of the pros and cons in the use of stem cells. So we need to go right back really to the beginning to understand what a stem cell is by looking at this particular picture here that I have in the center of the screen. This is a zygote. Now, a zygote is the correct scientific term for a fertilized egg cell. Now, that fertilized egg cell can become any number of things. It could specialize and become a sperm cell, could become a columnar epithelial cell. It could, as we've got in this third image, become a nerve cell or a neuron. It's going to help propagate action potentials and spread impulses. Or it could specialize and become a hepatocyte, a cell in the liver. The key to this is the fact that that zygote has the potential to become all of those four things that I've got at the bottom. Now, a stem cell is a cell that we describe as being undifferentiated or unspecialized. It as yet doesn't have or hasn't changed to have a particular role or function, if you like, but it has the potential to, and that's where the name totipotent comes in. The zygote or fertilized egg cell can mature into any type of body cell, so we call it a totipotent stem cell. So we're going to make a note of that word because that's really important. These stem cells are, and we'll put this down here, they are what we call totipotent. Now, totipotent, total, potent, potential. So these are cells with total potential to become anything. Now, once they differentiate and become specialized, they can no longer develop into other cells, so they lose that totipotency. So this zygote can become the sperm, can become a columnar epithelial, a neuron or a hepatocyte, but then that's it. It would lose that ability to specialize into something else. Now, usually once specialized, the cell can never become totipotent again. But it should be said that there are a few totipotent cells in mature animals, and it's those that are called adult stem cells. So those stem cells have three distinct general properties. And that's where we move over to the right-hand side. These three are key, fundamental things that these totipotent stem cells generally have. Number one is the ability to differentiate into specialized cells. That means cells with a job or a particular function. So the sperm will carry genetic information. The nerve, for example, as I said, will transmit nerve impulses. The red blood cell even, designed to carry oxygen within the circulatory system. The second fundamental thing that stem cells can do, or have rather, is the ability to divide and renew themselves infinitely. Because stem cells are quite early primitive cells, they can undergo mitotic division and divide very, very quickly and continually renew themselves. That's a little bit into why, for example, if we get a cut in our skin or an abrasion and it's damaged, our cells are able to repair themselves because actually in our skin, adult stem cells, for example, in the skin, will differentiate, specialize to become epithelial skin cells. And finally, third important thing to say is that these stem cells have the ability to relocate and differentiate where needed. So they're not stationary cells. These cells can go to where they are required ultimately. Now, there are three distinct types of stem cell. One of them is the totipotent cell that we mentioned before. And totipotent cells come from the zygote, and the cells are produced usually from the division of the zygote for up to about 5 days after fertilization. So you can take totipotent cells from that zygote after 3 or 4 cellular divisions, and they can become any cell. There are what's called pluripotent cells. So I think for this, what I might do is to shrink the screen here. So there's a bit of a nice board around what I've got so far. I'm going to make a note of these 3 because it's quite important. So the 3 types of stem cells we're looking at. So one are the totipotent, as I said, ones with total potential. The second type of stem cells are known as pluripotent stem cells. Now these are cells that come from the inner cell mass of the blastocyst. Now the blastocyst is the standard name for like the early ball of cells that we get after a number of mitotic divisions of the zygote. So that zygote will continually divide and grow, and it forms a blastocyst, a ball of cells. And if we take cells from the inner mass of that blastocyst, then what we've done is take some pluripotent cells. Now these can be isolated from embryos, and they have the potential to create every cell in the body except the placenta. So it's quite an unusual variant on the totipotent cell, but the pluripotent can become every cell except placenta. Then we have our third and final type of stem cell, and these are the multipotent cells. Now multipotent cells are the pluripotent cells that have become further specialized. So if you like, they're kind of a step on, if you think of it like that, a step on from pluripotent cells. They've become further specialized, and it's those that are often referred to as the adult stem cells. So embryonic stem cells are those that come from essentially the embryo, so pluripotent, or even earlier than that, the totipotent cells. But adult stem cells are those that have become further specialized down the way. And these can give rise to a limited number of other particular cells. So there is one argument, why not just use adult stem cells over embryonic stem cells for research, and it's because those adult stem cells have become further specialized than the pluripotent ones. So they're limited to what they can change into, if you like, or specialize into. For example, hematopoietic cells in the bone marrow can give rise to various types of blood cells, including red blood cells, white blood cells, and platelets. So there's a little bit about the types of cells, and what I'd like to do is just expand a bit more on why we don't use multipotent cells so much in research, like I said. Now, scientists have not yet identified multipotent cells for every type of mature body cell, and they're often only in minute quantities. Now, clearly, if these stem cells are in adults, what we've found, studies have shown that the numbers actually decrease with age, and it may take time to mature in culture to produce adequate numbers for treatment. Now, not just from aging, but also toxins and random DNA mutation, you can often find that these multipotent cells have DNA damage, and because of that, they're more limited in terms of their use in research. So that's just a little bit about multipotent stem cells. Let's think about how we culture them in vitro, so within a lab sort of setting, as opposed to in vivo using an actual body. So in vitro culturing of human embryonic stem cells, it looks a little bit like this. So we're going to shrink the screen down a little bit further again, just so I can outline this process for you. So we start this process, so this is a little bit about culturing. So we're going to start with a petri dish, if you like. And in that petri dish, I mean, this isn't by any stretch the best sketch, but it's just to show the procedure. So we have a petri dish here, And on that Petri dish we have that little dot. Now that little dot there is going to represent essentially our early embryo. So the early embryo is cultured in a nutrient medium. Then over time, if we were to look in that dish, let's do it from a bird's eye view. Also, it's a little easier to draw the diagram. So if we look at a bird's eye view, what we'll see is those embryonic cells will have started to grow and divide. And what we want, or what we notice rather, is that the outer layer collapses and the inner cell mass is freed from the embryo. So what we have in the middle here, this time, is our inner cell mass from that embryo. Chemicals, so I'm just going to draw a pipette here. It's my best attempt at drawing a pipette. I'll label it just. So using a pipette, chemicals can be added to break up the cell mass into smaller groups. So what we'll ultimately end up with is a plate like this, and in our culture medium you'll start to get groups of embryonic stem cells. I'm just going to draw in one more here. So we get groups of embryonic stem cells. So these are of, and I'm just going to put ES to mean embryonic stem cells. Now each group, what we find, will grow into a colony. So over time those groups, if we leave them, will start to, and I'll do it with just the bottom two, start to grow into a larger colony of cells. Now it's at that point where we can add what's called differentiation factor. Now special differentiation factors are added to the colonies in separate containers. So when we add differentiation factor, these particular chemicals, we can stimulate these colonies to grow. So from this plate that we have here, reduce the size again. So from this one plate that we have, we could extract a colony maybe for, I'm just going to use different colours. We could have maybe one colony that's going to become a colony of heart muscle cells. We could take one colony and culture it to form something different. So this could be, in fact, if we just move the screen across, this could be a collection of beta cells of the pancreas. These are the ones that secrete insulin. And we could get a colony of nerve cells, for example. Then once we have those different colonies being cultured in their respective petri dishes, we can transfer differentiated cells to damaged tissues. So if we had a person here, for example, and there were a variety of, let's say, medical conditions this person has, if there's a problem with the heart cells, so let's say they've had heart damage as a result of heart attack, we can take those heart muscle cells and introduce them to this patient. If we have damage to those beta cells linked to type 1 diabetes, again, we could take those and introduce those into a given patient. In fact, the potential uses of human cells from embryonic stem cells is so vast. I mean, if I just go through some examples. So Heart damage from a heart attack could be treated with heart muscle cells. Muscular dystrophy, a disease that could be treated if we're culturing skeletal muscle cells. If we cultured nerve cells, we could treat Parkinson's, MS, strokes, Alzheimer's, spinal injury, paralysis. So blood cells, let's say we were to culture some blood cells. Diseases that we could treat would be leukemia and blood diseases. If we've got burns or wounds, for example, with the use of stem cells, we could recreate or encourage the growth of skin cell. Osteoporosis is a disease that could be treated with bone stem cells. Cartilage cells could treat osteoarthritis, and even if we have damage at the back of our eye, so the retinal damage at the back of the eye, what's led to macular degeneration, it's called, with stem cells, we could replace those damaged retinal cells. Now, there are many arguments for and against the continued use of embryonic stem cells in research today, and I've outlined many of the pros that it could lead to cures for diseases, for these debilitating diseases that I've mentioned, and especially given the fact that IVF, and I've talked in another video about IVF numbers on the increase in fertility issues, spare embryos from IVF can actually be used instead of just being destroyed straight away, which in certain cases they are. So there are strong reasons why people really favour this kind of technology and are pushing it forward. But then some argue that the embryos which could have formed new life are simply being destroyed to get the stem cells, so they can come at it from a more sort of moral and ethical and perhaps in certain cases religious standpoint. Some argue to counter that embryos younger than 14 days can actually be classed as a human being, but it has been said that using stem cells to create human tissues could be a step towards doing whole humans. So we could end up almost replacing humans, have organs ready to order, if you like. It is, in any case, whether we agree or disagree, it will take many years before we know whether these diseases can be cured 100% successfully in this method. So it's still a very widely debated issue. But what I wanted to do in this video was just really talk about what stem cells are and general properties of them, which you can see at the top. Talk about the types of stem cells, so totipotent, pluripotent and multipotent stem cells, or the adult stem cells as I've referred them to, and to mention a little bit about how they're cultured and some of the reasons why we culture them. So some of the benefits of using stem cells and some of the drawbacks. Okay, hope all that helps.